Hey, thank you very much for inviting me to speak at the conference. I'm really sorry that I was unable to attend. We at CLL were very anxious actually to come. It would have been the first time that we were presenting our work at an autism conference. Unfortunately, that was not possible because of the security situation here in Israel, which led to a lot of airlines canceling the flights. So what I'd like you to do is really picture yourself 2 weeks ago. And 2 weeks ago, you're working on this talk, getting it ready. Now, Saturday morning, 6:30 in the morning, air raid sirens go off. You wake up, run with your wife to the room that your mother-in-law is sleeping in, which doubles as a bomb shelter. You close the door, you close the special window, and you wait. Sirens stop. You start to go out. Another siren. Siren stop. You have 2 or 3 minutes. Another siren. This goes on for 2 hours. Now you don't hear any bombs, so you think, okay, it must be a little not too close to us. But still, very scary. These sirens are very scary. Having to be in the room is scary. And on top of that, we're Sabbath observers, which means that we have to preset all our lights before the Sabbath. We don't use electricity that hasn't been preset. So this room is pitch black. So now picture yourself pitch black for 2 hours, not knowing what's going on, with your 95-year-old mother-in-law who doesn't even hear the alarm. That's the reason that it wasn't able to come today, because that attack led to a war, which led to the airlines canceling the flight. But I'm very happy that I'm able to give you this recording, and I'm sorry that I won't be able to get your questions online, but you can email them to me at the address below, fred.samuel@cll.com, and I'll be happy to address any questions you may have regarding this presentation. But what I'd like to do today is talk to you about the work that we've been doing to ask the question, can serum protein biomarkers identify children at high risk for autism spectrum disorder? And what I want to do is, first of all, go over very, very quickly, because I know you've heard it many, many times already in this conference, the prevalence of ASD. Then I'd like to discuss a little bit of what the state of the art is of the current diagnostic systems, not the things that are being developed, but how people are being diagnosed today. What are the limitations of those systems? And how biomarkers to be able to help. Why biomarkers? Then I'd like to get involved with CLL, what's the history of CLL, and how CLL is trying to develop biomarker assays, and why. What are the goals of those assays? Now, the current study, and the rest of that, I'll talk about our current study, its objectives, the methods we use, the results, and finally, what our future plans are. So as you know, and I'm sure you've seen this slide or versions of it many times in this conference, the prevalence of autism just keeps going up, up, and up. The last report that came out from the CDC, 1 in every 36 children born in the United States is autistic. That's almost 3% of the children. That's a tremendous number. It's a tremendous problem. It's a spectrum, so it's not just one disease, but it's a tremendous problem that has to be addressed, both on a diagnostic level, on a therapeutic level, on a school level, many, many levels. And that's obviously why we're all here at this conference. But how do we diagnose ASD today? And what are the limitations of those systems? First of all, we diagnose ASD using psychological and behavioral evaluations principally. Yes, there are people trying to develop tests like us, using blood, people using hair, people using urine perhaps, saliva, but none of those tests are approved. So none of them are part of the real standard of care today. And so children are being diagnosed by psychological and behavioral evaluations, various different tests. I don't have to explain them to you. And these tests are very subjective, and they require the child to cooperate. And usually they're not effective under the age of 2. Depending on the practitioner, they may not be effective under the age of 3. And that's big problem because I don't have to tell you that the earlier you get the children into therapy, the better those children will do in life. Excuse me. And if they have to wait 12 months, which is sometimes the case, from the point that they're evaluated by their pediatrician, referred to testing, specific testing for ASD, that's 12 months that they could lose. So there's a tremendous wait time in many different countries. There's also another problem that we discussed this with a number of KOLs, both in Switzerland and Canada and the United States, that about 30% of the children who are evaluated for ASD by the psychological testing, the results are inconclusive. And so these children are told to come back in 6 months, come back in 12 months. And again, you have even a further delay in when they're going to get treatment. So these children may not be treated for up to 2 years. 2 years when their brains are in a plasticity state, and they definitely can be helped by all the behavioral treatments that they've been given. And then the other aspect is that we know that ASD is a spectrum, and there are many proposed root causes for ASD, from the issues with the microbiome, issues with the immune system, issues from the mother, etc., and so on, genetic issues. But none of these tests that we do today can define what the root cause of this particular child's autism is. And so the only way we can treat is in a general way. So if we could develop tests, we had biomarker tests, for example, or genetic tests that would allow us to say, yes, this particular child has autism because of X. And now we could develop therapies just for that particular child. And that particular, so if we can have precision medicine methodology, we would have much more effective treatments. And finally, the follow-up of these children, in other words, people who get diagnosed and get treated in various ways, the only way you can follow up right now is by the psychological testing. But if you had biomarkers or ways to monitor the effectiveness of treatments, well, that I think would have a great help to drug development, because you don't have to wait weeks and months. You might be able to see the effect of the markers relatively quickly, and therefore see if you could mimic that or correlate that to the behavioral therapies, then you can use those markers, just like they're used for cancer, for example, to determine what the course of treatment should be. But that's lacking today. So with that as background, the fact that that's the state of the art, that where the how to treat the children is really up in the air in many times. I don't mean on a behavioral level. I mean on not just can you treat it in terms of the symptoms, but can you treat it and maybe correct the syndromes, right? That's missing, and that's where Cell-L was initiated. Cell-L was founded by Dr. Benny Gesundheit, who's a pediatric oncologist. And Benny was working at Hadassah Hospital in Jerusalem as a stem cell expert in the department that was treating children for various ailments, and children and adults, but in his case, children, he's a pediatrician, with stem cells. And some of the children that were being treated by that department were autistic. And they saw that some of them were being helped by the stem cell treatment. So Benny, who was not involved directly in those initial studies, decided that he wanted to learn more and more about autism. He says he spent a year in the library and then wrote a review article about autism with some of you were co-authors on that paper. But he then set out as a mission, and his mission was to improve the outcomes and the quality of life of the children with autism. How are you going to do that? Well, you have to develop treatment, but you also have to develop diagnostics, be able to pick them up earlier. Maybe you can treat them early. So the second mission is to provide opportunities for earlier intervention by changing the ASD screening paradigm using precision medicine. Identify the children, as I said before, what particular treatment can help which particular child. That would be the ultimate end game of Cell-L's mission. Now, as I mentioned, Benny went to the library for that year, and he wrote a review article on autoimmunity and autism spectrum disorders. And also, as I mentioned, some of you were co-authors on that, but the bottom line and the thing that I want to point out is that the hypothesis proposed was that there were maternal cytokines, antibodies, T cells, perhaps, that pass the placenta and go to the immature fetal brain that's being developed. And they impair the development of that brain, and that leading the child to be autistic. And the other part of the theory is that those cytokines and antibodies, etc., would still be found in the autistic child, so that you could potentially screen for them and know that this particular child's root cause of his autism, his etiology for his autism, is an autoimmune one, and then treat him on that level. And that's really the background to Cell L, trying to develop, ultimately wants to get involved perhaps in treatments, but right now wants to identify the children so you can figure out which treatments to use. So the goals, as I said, the goals of our projects, and I'm talking to you right now on really only one of them has begun, which is the first one, which is to change the ASD diagnostic testing paradigm, develop laboratory biomarker assays that can assess infants and other older children. The studies I'll talk about today, we're testing children from 3 to 12, and we've initiated some studies now with infants to try to extend the findings. But longer term, we want to look to see if we can identify the root causes of ASD using biomarkers. For which children, as I said earlier, using those precision medicine techniques, which children have autism based on which root causes, and how can we then treat them? Then can we monitor those treatments also using laboratory biomarker assays? And finally, can our tests be used to differentiate ASD from other neurodevelopment diseases? But as I said, right now, the focus is on that first development of the first test, developing a laboratory serum-based biomarker test. And the goal of this study is, as I said, to establish an objective serum biomarker test, focusing on proteins associated with the immune and the inflammation system, to identify children at risk to develop ASD versus those typically developing. The methods we use, first of all, we had to have Helsinki approval. That we have from Kari Tzedek Hospital in Jerusalem. It's a multi-center test, so we have collected samples mostly from Israel, but also from Canada. The blood was collected in venous blood collection, preparation of serum from ASD and TD children. The collection points, we went to the children, we go to the children. So we collect the blood in schools and hospitals and clinics. We don't wait for them to come to us. We even go to their homes. And with that, we have a cohort. We collected 102 ASD blood, 97 from TDs. Once we had the serum prepared, we sent it to our research partner, Ray Biotech in Georgia, who have a multiplex sandwich ELISA-based array platform. They then sent the raw data back to us, where our statisticians were able to do statistical analysis to identify the best performing algorithm. Now, some proteins may go up, some may go down, and that's part of the algorithm. But first of all, we had inclusion and exclusion criteria. Children in this particular study were aged 3 to 12. The ASD children were documented as having ASD using psychological testing. The TD children had no signs or family history of ASD. And of course, we had signed informed parental consent. The exclusion criteria were that we did not want to, because we're looking at inflammation and immune response, we did not want the children to have had any mild infections even in a month prior to taking their blood. No colds, no fevers, no need for antibiotics. We didn't take any children that had severe convulsive disorder. And anyone who had a serious infection or use of systemic steroids or cancer treatment in the preceding 6 months were excluded from our study. If you look at the demographics of these children, for the ASD children, they were about 90% boys. For the TDs, 60% boys. The average age for the ASD children was around 5, for the TDs closer to 7. And our statisticians did the analysis to see if there was any bias based on that, the fact that there were these changes, that most of the ASD children were boys versus the TDs The TDs were slightly older because parents weren't just volunteering to bring their very young children into us. So they're a little bit older. But they came to the conclusion that there was no statistical bias, there was no issue, and so we could use the data as a agglomerate. As I mentioned before, most of the children are from Israel, some from Canada. When we sent the samples to Ray Biotech, they used their system, which they refer to as a human kiloplex quantitative proteomics assay, or Ray. It's a multiplex sandwich-based ELISA-based assay. They put sandwich assays for 40 different proteins on every chip. Up to 1,000 markers were studied. Each sample was run in quadruplicate. They then got image analysis of the scanned chips. They calculate, but you get standard curves. They sent us that raw data, and as I mentioned before, our statistical team then built an algorithm based on their analysis. And we were able to identify 13 biomarkers. Some go up, some go down. The first 8 you can see have been reported to be in the literature to have potential to be biomarkers for autism. The other 5 were unique to our study. And we were very, very happy with the results because we got 87% sensitivity. Now, I want to remind you that in every diagnostic, there's a question of what's worse, a false positive or a false negative. And for example, if you were to be doing an HIV test, well, false negatives would be the worst thing that could happen. That person would go out and infect other people. But if you're doing a rubella IgM test, for example, and if that pregnant woman has rubella and IgM, well, you might abort her baby. So if in that case, a false positive would be the worst. She's lost her baby when she didn't need to lose it. So every diagnostic, you as a developer, you gear it one way or the other. In the case of autism, I think it's much better to have as few false negatives as possible, because in the worst case, the false positives will be given therapy, and the therapy is, in most cases, behavioral therapy or speech therapy, and so it's not that it's not intervention in terms of drugs, etc. The other thing to point out is that in our case, the sensitivity of 87%, quite remarkable, because that suggests that the immune and inflammatory systems are involved in almost all the autistic children. It could be that the 13% that we miss who are diagnosed as autistic are because their root causes does not result in that. They are autistic, but we don't have the tool to pick them up with our tool, because their root cause does not affect the immune system or the inflammatory system. But overall, we were very, very encouraged by these results. And so what are our future plans? Well, we've collected a new cohort of 160 ASDs and 160 TDs. We're going to test them using a different microwave system, a different partner this time, and try to confirm that the initial markers are correct, but add to the strength of our assay by trying to find other potential biomarkers, so that we want to get to at least over 90%, preferably accuracy, but certainly sensitivity. And then, from once we have identified those markers, we'll develop a custom array using only the markers that we've identified and used in our algorithm, and then perform independent validation studies. Some will be blind, some will be retrospective, but they'll be done by other laboratories, not by us. Finally, that we'll complete the development of the final product and then go for regulatory approval, and then ultimately to commercialize the test. We expect that this process will take us about 3 years. So I thank you very much for your attention. I wanted to point out the team at Cell. I won't go through all the names. I mentioned Benny before, and I just want to point out Charlie Pico, who was here last year. Some of you may remember him. He's the head of our business development. And now I thank you very much again for your attention, and we'll be happy to receive any questions. And again, apologize that I was unable to attend.